any questions about anything we covered so far? Alright, so I moved the test, you guys may have noticed, on Canvas to the 6th from the 4th. So, the plan is that we'll probably finish half of Chapter 9 today. We'll finish the rest of it on Thursday, and then we'll do a review on Tuesday, and then you'll have your test on that Thursday. Alright? So, even though the syllabus says the instructor reserves the right to move dates or whatever, it's a conflict with somebody, come and see me and I can put it in the testing center on Tuesday, and you can take it then, or Wednesday. It's up to you. Doesn't matter to me, because I'll have it made up by then. Alright? So, same drill. It's going to be multiple choice, 50 questions. Bring a green Scantron and a number 2 pencil, periodic table, and you can bring a calculator, non-programmable and non-graphing. And I replaced my 50-cent calculator. Look at this one. I got this for $1 at the dollar store too. Yeah, look at that. It does. It's sine, cosine, tangent, natural log, logarithm, hexadecimal, binary, rounding. Yeah, $1. Alright, so we left off talking about, you guys know, the timeless wonder of it all. Sunlight falls on the plants and they magically do photosynthesis and make sugars. And those sugars are linked together in long chains, the glucoses, and that makes amylose, right? And so you guys, you eat the food, the sugars, and then you put it in your mouth, and it gets digested by that enzyme. You guys remember what that enzyme is in your mouth? Amylase, correct? And so then you swallow your food. So let's say this morning you woke up, right, after a night of fasting, so you had breakfast, which is what it comes from, and you ate at least carbs, right? So you swallow it. What does amylase produce, by the way? Maltose, good. So you swallowed the maltose, and in your stomach, the acids in your stomach can only break those glycosidic bonds between the glucose molecules. So what do you end up with in your small intestines after it goes through your stomach? It can only break glycosidic bonds. And the crickets. So it's glucose molecules, right? Because that's what links the maltose together. And so then that goes in your small intestines. And so now you have a lot of glucose in your small intestines. Do you think you have a lot of glucose in your blood? You just woke up, slept all night. No. So where's the area of high concentration for glucose? In your intestines, and you need to get it into your blood, so it's going to go into your blood, right? And what kind of transport is that? No? When we talked about, we have passive and active. Passive, so do you think it's going to need a channel, or can it pass through the cell membrane by itself? Right, so what do we call it if it needs a channel? It is active, but there's a specific name for that. It's called, it starts with facilitated and ends with diffusion. So now you have the glucose in your blood, right? And too much of a good thing is toxic, so you don't want the glucose to hang out in your blood too long. You can ask a diabetic. So that activates your pancreatic cells that produce insulin, and then insulin binds to the cell membrane. And that sends a signal to the nucleus to make the proteins, to make a channel for glucose to go into. So what do you think the glucose is highest, in the blood or in the cell? So then, is this active or passive? And it goes through a channel, so what's it called? Yes, facilitated diffusion. Good. So, this is where we're at right now. With the glucose molecule, and where is it at? What do we call this part of the cell? The cytoplasm or the cytosol. Alright? So I just took you through your breakfast digestion. Now there's two things that can happen from here. We can have fermentation or we can have cellular respiration. So the only difference is that in fermentation there's no oxygen present. So if you hold your breath, eventually when you run out of oxygen in your blood, your body, your cells will be forced to do fermentation. And if you continue to breathe, then your cells will do cellular respiration. Now, the main difference between the two is glucose is still broken down in a process called glycolysis, but during fermentation you make 2 ATP, and during glycolysis in the Krebs cycle and electron transport, which we call cellular respiration, you make 36. That's what you net. So, do you think you could live off 2? All you have to do is hold your breath and see how long that lasts. Right? So you need to breathe to generate that extra 16 times more energy than you would without it. Alright, so fermentation produces ATP, but it produces a small amount of ATP. It's done in the absence of oxygen. It results in the partial degradation of the sugars, in this case glucose, and then again it only produces 2 ATP. Respiration, on the other hand, and we've done this, we've written out this formula lots of times. Glucose C6H12O6 plus oxygen, and that's a 6, right? Yields carbon dioxide and water, and then you get energy out of that in the form of ATP. You can do any of these. You can use any kinds of fuels to produce ATP. All you have to do is look on our nutritional label and see what part of that food produces calories. So what are the common calories from? Carbs are one. What else? Fats, proteins, sugars are carbs, right? So remember, they're simple and compound, but they're all carbs. Even fiber is a carb, right? But it's non-digestible. What kind of sugar is fiber? Cellulose. Excellent. Alright, so you can use any of this, but the pathways we're going to focus on are the glycolysis ones, the ones that break down the sugars. Alright, so the first thing that's going to go on here is that we have to transfer electrons. So we take electrons off glucose and we put them on these electron carrier molecules. When we have a transfer of an electron, that reaction is called an oxidation reduction reaction. So it's a fancy name for transferring electron from one molecule to another. Does that make sense? So this is an example of, this molecule A gets an electron, gives an electron to molecule B. Now molecule B has an electron on it, right? And molecule A has one less electron. So if you gain an electron, then you're said to be reduced. And if you lose an electron, you're said to be oxidized. So the way I remember it is Leo the lion goes grr, right? So lose an electron, you're oxidized. Gain an electron, you're reduced. But you can remember it however you want. You want to memorize it, that's fine. But you need to know that because that's how biologists and chemists speak. We talk about this molecule's oxidized, this molecule's reduced. And if you don't know what oxidized and reduced means, you won't be able to follow the electron. Okay? Any questions?
What was that you said?
So, which one? The trick to remember it?
I don't.
Yeah.
Leo, the lion, goes grrr. Leo, lose an electron, oxidized. Grr, gain an electron, reduced. So you lose an electron, you're oxidized, gain an electron, you're reduced. Alright, so this is actually what's going on. I don't care that you guys memorize the shape of these molecules. If you take biochemistry, they'll probably make you learn this, but you're not in biochemistry, you're in general biology 1, so I'm not going to make you learn it, but you should recognize this molecule. What sugar is this? It's a pentose, that's correct. It's ribose, that's right. And you know it's ribose because it has two OHs there. If it was deoxyribose, this would just be an H. And then this is the base, A, right? And so this would be something that, so if DNA had a T in it, when RNA was made, this would be the molecule that's put to match it, right? T's match to A's. But this is what we call a dinucleotide. So it has two, but this one you've never seen before. It's called N, and it's not used in RNA normally. It's used to carry electrons. And it's a good electron carrier because if you notice this N is positively charged, this nitrogen. You see that? And so what happens is that you have two hydrogens that come from glucose. So what are two hydrogens if we break them down into their subatomic elements? What are they made out of? Subatomic particles. So do hydrogens have neutrons? What's hydrogen's number? What's its mass? So no neutrons. It has one proton and one electron. So if we're talking about two full hydrogens, we have two H pluses, which is a proton, and two E minuses, which are electrons. Can we follow that? Okay. So it's pretty simple. The hydrogens in the glucose molecule get transferred to NAD plus, which stands for nicotinamide. I don't care that you know that word, but you need to know that NAD plus, because it's a very important molecule. So one of these electrons goes on to the N plus, so that makes it electrically neutral. So I'm going to erase one electron. You guys follow where that went? This is N plus, and this is N that doesn't have a plus. So if you're plus and you get an electron, what are you? If you're an N plus and you gain an electron, what are you? No, you're neutral, right? So this is where one electron went. Okay? All right, believe me. Here's a hydrogen, right? And magically, here's another hydrogen. What's a hydrogen made out of? One proton and one electron. So now I can erase the electron because that's part of the hydrogen, right? And I can erase one of these protons. So what's left over? One proton, right? Go back and watch it again. That's right. It's not a magic trick. It's right here. So two electrons, two protons get onto this molecule, and so it becomes NADH because now it's electrically neutral, it's not positively charged, and it has a hydrogen added to it. Alright, so NAD plus, because of this reaction, becomes NADH because we've added two hydrogens plus an H plus. What? I might ask you which one. So NAD plus, is it oxidized or reduced in the reaction to make NADH? So is NAD plus oxidized or reduced? It didn't lose the electron, it gained the electron. NAD plus got an electron added to it to make it neutral. Right? Believe me? It's true. So the electrons came from where? Where do the hydrogens come from? What part of the food? Glucose. That's right. It's written right there. So the glucose was oxidized or reduced. The glucose is oxidized and the NAD plus is reduced. That's how biologists talk. So, use it in a sentence, oxidized, reduced. Go home, practice on your friends. Write something about it on Facebook. Tweet about it. Alright. Whatever you want to do, it doesn't matter. The molecule that does that is called dehydrogenase. What's a dehydrogenase? Remember we did this in lab? What's its job? What's D mean? And what's hydrogen? There you go. So it took off hydrogen from where? Glucose, and put it on the NAD plus to make NADH. Excellent. So NAD plus, or NADH, is an electron carrier. This is really important because have you ever been electrocuted? You haven't? Oh, you're so lucky. I have several times, like remodeling, stuff in my house. It's not pleasant, right? Nobody likes to get electrocuted. You don't hear people lining up going, yeah, let me stick that fork in an electrical outlet. And that's because it has a profound effect on your body and your cellular structure. You don't want electrons flying around inside your cell any more than you want them flying around inside your body. So you want to be able to move these things safely. This molecule's job is to do that. If you want to, think about it as like a UPS truck that someone stuck an electron in. So now you can ship it around the cell without having to worry about where that electron is going to end up. Okay? Alright. This just basically says the exact same thing that we just talked about. So 2 electrons, 2 protons are transferred onto this molecule to make NADH, and hydrogen's protons are a byproduct. Alright, so in the end, ultimately what we're going to do is combine hydrogen and oxygen. So, do you guys know what fuels the space shuttle? Hydrogen and oxygen. So what would happen if you took all the food that you ate and suddenly combined it together from the hydrogens or the glucose and the oxygen that you breathe? You would explode. So that wouldn't be good, right? So the reason that we have all these little steps, all these enzymes, remember that massive chart I showed you for the metabolism, is because you do this in little baby steps so that you don't get a massive explosion. They're just little tiny ones that don't affect you that much. Alright, so that's why there's so many enzymes involved in this process and so many steps as well. Because you don't want to burst into flames. That would be bad. Alright, so that brings us to the heart of the process, which are the 3 major steps in breaking apart sugar and releasing the energy from it. So remember the formula: glucose and oxygen make water and carbon dioxide. There's nothing different from each side of that equation as far as molecules, right? So what's the difference between the two? They're rearranged differently, right? So if I gave you a model of glucose and a model of oxygen, O2, and asked you to make water and carbon dioxide, you could do it, couldn't you? But what was the first thing you'd have to do? Well, you don't have to remove water, but I mean physically, what would you have to do if I gave you the model? Yeah, you'd have to take it apart. So the first thing that we have to do is we have to take apart the glucose. And so the first step is called glycolysis. Glyco, do you remember what that word means? it means sweet, right? So it refers to sugar. And remember what lysis is? Well, yeah, it's to break apart. Remember we talked about plasmolysis, when you rip apart the plasma membrane from, what's that, hypertonic or hypotonic solution? Alright, make sure you guys know that in 10 days. Alright, so, and the answer is hypertonic. So glycolysis is the first step. We're going to break apart glucose into 2, 3 carbon sugars and rearrange those into a molecule called pyruvate. Every one of these steps, I don't want you to memorize all the stuff that's involved because there's a lot. And again, that's part of a biochemistry class. But what I want you to know is what goes in and what comes out. So that's important. All right? And then this occurs in the cytoplasm. The second step is the Krebs cycle, which occurs in the mitochondria. And I showed you guys that in lab when we did the cell fractionation lab. And then the last one is the electron transport chain. And that's in the inner membrane of the mitochondria. So we're going to go over each of these in detail. Any questions so far? Alright, so glycolysis occurs in the cytoplasm or the cytosol. Glucose is converted to pyruvate. The pyruvate goes into the mitochondria, into the matrix where the Krebs cycle is found. This makes 2 ATP, this makes 2 ATP, and then this makes 32 ATP. Alright. So we're here, right? This is where the enzymes are that are gonna start breaking apart the glucose. Like I said, it splits it into 2, 3 carbon sugars. They are then oxidized. So what does that mean? Leo the lion goes grr. Lose an electron, what are you? So these are losing electrons. Remember I told you the glucose loses the electrons, and where do they go? NAD plus to make NADH. Excellent. And then they're rearranged into 2 pyruvates. It's just another molecule, right? I mean, you guys know what water's formula is, and you know what glucose's formula is by now. It's just another molecule with another formula. So it's nothing fancy. It's just a molecule, but it still has a lot of energy in it. The process still retains tons of energy because you're only getting a couple ATP out of this. So we still want to take the energy out of the pyruvates, and that the Krebs cycle and the electron transport chain does that. They're all enzymes, specific enzymes. I'm not sure if the video works because they updated to PowerPoint 10 or 2010, but we'll see. So one thing that's important to remember is it doesn't matter if you're breathing or you're not. So in the absence of oxygen, in the presence of oxygen, glycolysis happens. Right? You could get a bumper sticker that says glycolysis happens. Then you'd be really nerdy. Alright. Then people will be like, what? Yes. This is a real word. Alright. So, 2 phases of glycolysis. One is the energy investment, and one is the energy payoff. So it's like the rock rolling downhill, right? You got to put some energy into it to get it to start moving. Once you've got it to move, you can get energy out of it. All right, so glycolysis costs 2 ATP, but the payoff is you get 4 ATP. So if you gave me $2 and I gave you $4, you'd do that all day, right? It's a pretty good deal. What are you getting out of it? If you give me $2 and I give you $4, what are you getting out of it? So when we talk about net gain of ATP, our net gain is 2 ATP, right? Because it costs 2, but we get 4 back. Does that make sense? All right, so make sure you guys know that. The energy payoff, we get 4. We also create 2 more NADHs. What are these for? Their job is to carry electrons safely through the cell, right? That's all they do. They just carry electrons. They're UPS trucks with electrons in the back of them.
So NAD plus and NADH both just electron transport.
NAD plus doesn't have an electron on it. It's an empty UPS truck. NADH has an electron on it, so it's a full one. So only NADH has an electron to carry. NAD plus doesn't have it. And then we get 2 pyruvates. Alright, so remember I told you guys for the test you need to know what goes in and what comes out. So what goes in to glycolysis? Excellent. So let's just keep a tab here. What else goes in? Yeah, glucose. And then what comes out? 4 ATP. What's this for? Carrying electrons, good. And then what else? So pyruvates still have energy in them. They're molecules that come out of the cycle. So in fermentation, they get converted into alcohol, ethanol, if you're talking about yeast. For you guys, it would be lactic acid, which is toxic to your muscles, so that's what causes you to cramp. But if you're breathing, it goes through into the mitochondria. You can take more electrons off of it and make more energy. Alright, so this is how it works on a molecular level. Again, don't freak out. I don't want you to know or memorize these molecules. There's only a couple things that you need to know. I'm just showing you what exactly is going on here. So here's glucose. Remember hexokinase? I showed you that from the last chapter, that blue molecule. Probably forgot, but it's okay. Hexokinase, what's a kinase job? I told you that's the one enzyme you need to know. What does it do? Adds a phosphate, that's right. So look what it did. It added a phosphate, just like what it's supposed to do. Where did the phosphate come from? ATP. So now we have ADP. And so now this molecule is called glucose 6-phosphate. Why is it called that? The phosphate's on the 6th carbon. That's it. So here it is, right? And this is called phosphoglucoisomerase. It's a big word. What does it tell you? It's going to create an isomer from what? Glucose with a phosphate. So it reacts with this is a glucose with a phosphate, right? And so what's an isomer? No, that's an enantiomer. Isomer, it could have a different shape, right? But the same parts, right? Iso means same and meros means part, same parts. So fructose is an isomer, right? It's still C6H12O6. And so this is called fructose 6-phosphate because now the phosphate's on the 6th carbon of fructose. That's all it's done, right? This enzyme is called phosphofructokinase. What do you think it's going to do? What's a kinase do? Add a phosphate to what? Fructose with a phosphate on it. Well, guess what? There's fructose with a phosphate on it, so it's going to add another phosphate. Where's the phosphate going to come from? ATP. So now we have two phosphates, right? On the opposite sides of this molecule. Now, if you wanted to split this molecule in half, what would you have to do? Well, no, this isn't a... Huh? Break the bonds, right? You got two phosphates on either end. What do those want to do? What's the charge on a phosphate? So, if I put two negative magnets next to each other, what do they want to do? Break the bonds, right? So that's what it does. It splits this molecule right down the middle. So we've taken a 6-carbon sugar and turned it into 2 3-carbon sugars. What's a 3-carbon sugar called? Didn't know. That's a fat. Triose, right? Sugars end in OSE. So we made 2 triose from a... What's a 6-carbon sugar called? Hexose. Excellent. Now, there's an isomerase, so you can look at this and count all the carbons and oxygens and hydrogens, but I promise you this has the same chemical formula as this. So it's going to convert this into this. So what comes out of this is 2 glyceraldehyde 3-phosphates, which we call G3P. You're going to see this again because this is what plants make during photosynthesis. They don't make glucose directly. They take 2 G3Ps and they run this in reverse. So if you ran this in reverse, what would you end up with? Glucose. That's how plants make glucose, and then they link it together in long chains, and then they store it as long chains of glucose are carbs. Excellent. Right? And then you go and you sneak them on the plant and you eat it, and then you do this process and break it apart. Everybody okay? Oh, this one is converted. The isomerase converts it into G3P. Yeah, so 2 glyceraldehyde 3-phosphates. So in your book, instead of the artist drawing 2 of the exact same things, he just put the number 2, or she put the number 2 in front of this. And so that just means that there's 2, right? Because we made 2 G3Ps, so all of these are doubled now, all these products. So 2 NAD pluses make 2 NADHs, which is right here, right? So we paid in the 2 ATPs, and now we've got back our 2 NADHs, and then we're going to take 2 phosphates here, so that's 2 of these, right? And then again 2 phosphates here, so that's 4 total. And then what comes out at the very end? 2 pyruvates. So, like I said, I don't want you to memorize or know this. I'm just showing you that it's not just some magic wand, right? There's a lot of junk going on to make this. This is what goes in, and this is what comes out. But there's a lot of enzymes that are doing work in there to make this happen. You don't need to know them all. The only thing you need to know is a kinase, right? I'm going to ask you on the test. I'll tell you straight up. I'm going to ask you what a kinase is, because most human diseases are caused by failures in kinases. So it's kind of important. Just saying. Oh, it works. So this is exactly what's going on. Hexokinase comes in, makes a glucose 6-phosphate, fructose 6-phosphate. Now we have the 2 phosphates, right? The molecule gets split in half. Isomerase rearranges it. So now we have 2 glyceraldehyde 3-phosphates, right? And that cost us what to make? 2 ATP, right? That's the energy investment. Now we get the payoff. So we remove the hydrogens to make NADH from NAD plus. Remember, the byproduct is the proton. So we've got two NADHs here, and then we take off the phosphates and we get two ATPs. And some more rearranging occurs, and water's removed. So remember, we talked about water is the product of the reaction. And then we get two more ATPs. So we got our 4 ATPs back. And then what molecule is that? It says right there. All right, I'll read it to you. Pyruvate. Since I'm getting no audience participation tonight. So that's only a little bit of the energy, right? There's still a lot of energy left. And so what's the powerhouse of the cell? The mitochondria, right? And it's that for a reason. So all this is taking place in the cytosol or cytoplasm. And now we have pyruvate, and we have to get pyruvate into the mitochondria. So how many membranes does the mitochondria have? 2. And so when we put pyruvate into the mitochondria, there's lots of pyruvate. Remember, your body has to generate 10 million ATP per second per cell in a working muscle. So there's a lot of pyruvate getting shoved in here. What do you think the buildup of pyruvate is going to be? Where's the concentration going to be greatest? Inside the mitochondria or out here? Inside, and you'd be right. So we want to put this inside. Is that with or against the gradient? Against the gradient. So how do we get it in there then? And what do we need for active transport? That's exactly right. What do we use for active transport? ATP. So we have 2 pyruvates. How many ATP do you think it's going to cost us to get it into the mitochondria? That would be right. So here we netted 2 ATP, right? But now we lost them by the next step. So if we say this is the first step, then the second step would be moving pyruvate into the mitochondria. All right, so what goes in here?
Is that why glycosis only makes 2 enzymes that process?
No, because if you're not breathing, you get to keep those 2 ATP. Pyruvate only gets transported into the mitochondria in the presence of oxygen. So hold your breath. When you get to the point where you're about to pass out, you're probably going through fermentation. Or, if you're running, like exercising, you can only take in so much oxygen, right? So your body is forced to do fermentation. And in that point, you get to keep the 2 ATP. But if you're breathing air, you don't get to keep it. It's a good question. So what goes in here? Good. And? Excellent. And so there's some chemical reaction. There's enzymes, right? There's always enzymes. They're doing these jobs. They take off this carbon, oxygen, oxygen. So what is that? Molecular formula for a carbon with 2 oxygens. Yeah, what do you breathe out? What's the product of the reaction? So this is where the CO2 that you're breathing out right now comes from. Wait, see that? So CO2 is given off. We make NADH. Remember, we have 2 pyruvates, so how many NADH do we make in this? molecule that comes out of this, it's another molecule, right? It has a different name because it has a different shape. But it's called acetyl-CoA, and it is formed by coenzyme A. What's a coenzyme? What's a cofactor? So a coenzyme is an organic one, and a cofactor is an inorganic one. All right, so this could be a vitamin or something, but it's not. It's a specific molecule called coenzyme A. But that's its job. So if I ask you on the test, there's this enzyme that requires a coenzyme to react. If the coenzyme isn't present, will it work at all? Will it work? Not even a little? Just a tiny bit? You're right, it doesn't work at all. So that's important to remember. So everybody got it? That's what goes in, and that's what comes out. This is what you need to know. You can memorize it, but I want you to understand what's going on here. And you'll see this again, I promise. You'll see this again in anatomy. You'll see this again in microbiology. You'll see this again in genetics. All right, so step 2, now we're on step number 3. So the next step is the Krebs cycle. So now we've taken the pyruvate and we've made it into acetyl-CoA, and that is inside the inner part of the mitochondria. What's that called? Chapter 6. That's right. It's the matrix. So this is where we're at now. Matrix of the mitochondria. So what goes in? Two acetyl-CoAs, right, from our original glucose. Remember, it's just showing you one, but we have two still. So acetyl-CoA combines with a chemical called oxaloacetate. And that produces citrate, which is also known as citric acid, right? It's the same stuff that's in your soda can or Sprite or whatever. So the Krebs cycle is called a cycle because what it starts with is what it finishes with, right? So you see that oxaloacetate and acetyl-CoA combine to make citrate, and that goes all the way around, and what do we end up with at the end? Oxaloacetate again, right? So as long as you're feeding it acetyl-CoA, the wheel keeps spinning. It's not a real wheel, right? There's not a circle in there, but it's easier to draw it that way so you can see that it's regenerating, right? It's recycling. In fact, that's where the word came from. This is a cycle, right? It's recycling. All right. So, what comes out of this? What's the purpose of this? So water's used, right? So what comes out here? So how many CO2s? 2, right? But we have 2 acetyl-CoA, so how many total? So remember the formula? How many CO2s do we give off per glucose? Remember, it's C6H12O6 plus 6O2 yields 6 CO2, right? So we have 4 plus 2 is, that's what you're exhaling right now. So what you're breathing out, the reason you're breathing in air is for the electron transport chain, but what you're breathing out is byproducts from glycolysis and the Krebs cycle. What else are we making here? Our friend the electron carrier, right? How many of these? 3, but remember there's 2, so 6 NADH. And then there's another molecule that you haven't seen before called FADH2, which is also an electron carrier. The only difference is this thing holds its electrons more tightly, so it's harder to get the electrons off of this than it is to this one. FADH2 is more electronegative. And then what else? What's at the bottom? Right. How many ATPs? All right. So at this point, what have we totally gotten out of this? So how many ATPs? So we put in 2 to get 4, so we netted 2, right? But we use the 2 to get the acetyl-CoA into there, so we have 0. So do we make ATPs over here? Do we make ATPs in Krebs cycle? Yeah. How many? 2. So all of this we've made 2 ATP. That's the same as holding your breath, right? So is that going to be good enough to live? Hold your breath and find out. So what else did we get out of this? Carbon dioxide we know is a byproduct. I'm just showing you that. You exhale that. You're not using that for energy, are you? So what do we have left? All we have left are the electron carriers, right? We have NADH. How many of those? Two, 6, 2 plus 2 plus 6 is, and then our other electron carrier, FADH2. This is all we've got. So where's all the energy? Where are we going to get the rest of these ATP that we need to live? From the electrons, from the electron carriers. That's right. So the next step is the electron transport chain. We have to move the electrons just like you would for electricity, because moving electrons can do work. You guys know that, right? Your phones work off of it, your refrigerator, your vacuum. Like, go home and shut off your electricity and see how many things that you rely on electricity for. Someone said no. Alright, so this is just showing you exactly what we did. We went through each one of these steps. So glycolysis, we get 2 ATP and 2 NADH. Pyruvate, moving it into the mitochondria, we get 2 more NADH. Krebs cycle, or it's also known as the citric acid cycle. So Krebs, the scientist Krebs claimed he was the first person to discover it, but some other inventors said, or scientists said, no, I was. And so now it's commonly called the citric acid cycle, because what's made from combining oxaloacetate and acetyl-CoA, do you remember? Citric acid. So that's why it's called that. So 6 CO2s. But the important thing is that this is where all the energy is. It's in the electrons that are being carried safely through the cell. Because you don't want electrons flying around in your cell. That would be bad. Free electrons damages your DNA. It causes aging. It makes free radicals. It's nothing but bad stuff. So you don't want electrons flying around inside your cell. And then 2 ATP. So the rest of the energy is in these electron carriers. And once we put these in the electron transport chain, we're going to get 34 more ATP out. So if we get 34 out of the electron transport chain and we get 2 out of the Krebs cycle, how many total do we get? 36, right? But what if I told you bacteria make 38? Are they better than us? Why do they make 38? That's exactly right. They don't have mitochondria, so they don't have to pay. They don't have to pay these 2 ATP. They get to keep them, because what they use is their cell wall as the outer membrane of the mitochondria. So they're going to pump protons like this, whereas we have to use the mitochondria inside the cell. There. And then you take antibiotics, it does this to the cell wall, and then what happens to the bacteria? Why do they die? They can't make ATP, right? You can't make ATP, you can't live. So that's why antibiotics work. Does that make sense? And they don't affect you because you don't have a cell wall, you have mitochondria. Alright, so to understand the electron transport chain and how this works, you have to understand how the mitochondria is laid out. So remember, we have our cell, this is the cytoplasm, and the mitochondria, we have an outer membrane, and we have an inner membrane, and then what's the center part called? Perfect. And the matrix is folded back and forth. Those things are called cristae. Why would it do that? What's the purpose of those folds? It's more surface area so you can put more electron transport chains. So, because believe me, if it wasn't like that, there's no way you could make 10 million ATP per second per cell. Alright, so I'm going to take you on a little trip back to 1970, and scientists were just now starting to discover that the mitochondria were responsible for making ATP. They did it the same way we did. They did cell fractionation. And they looked at it under the microscope. They noticed that these chemicals inside the inner membrane of the mitochondria that they called cytochrome. So what's chromos mean? Color, right? So they noticed that they changed color. These proteins changed color, and they changed color based on whether they were oxidized or reduced. So if they were oxidized, they were red, they emitted a red wavelength, and if they were reduced, they emitted a green wavelength, right? And they could detect that. They used the spectrophotometer just like we use in the lab. And they were like, well, that's interesting, because what has to happen if something is oxidized and reduced? So electrons have to be moving through this thing. So that clued them in on that somehow moving electrons were generating ATP. So they started ripping the cell apart, right, the mitochondria, and looking at all the proteins that made up the inner membrane, because they knew that's where this was going on. They starved the cells from oxygen. What do you think happened? They didn't make ATP, right? Yeah, they had to ferment. So after some research in early 70s, they figured out that the electrons were coming off of NADH and FADH2. Those electrons were getting passed onto the cytochromes, making them change colors, right? And then eventually oxygen was the electron acceptor. Why oxygen? Why do you think oxygen is at the end of this chain? So if you're going to get an electron on you, what do you have to be? Yeah, so what's the most electronegative element besides fluorine? Oxygen. Fluorine you can't reverse. Remember, we have to have the delta G imbalanced. So if fluorine gathered all the electrons so you could never get them back off, you couldn't use that. It would make everything zero and life would stop existing. So that's why we breathe oxygen, because it can take the electrons. And guess what? When you stick a fork in an outlet, what happens to the electrons from the outlet? Where do they go first? A metal fork. And then where do they go? And why do they go into you? What do you think's more electronegative, the fork or you? Why? What are you mostly made out of? How much? Do you remember? No, that's water. 65%. Remember I showed you the second chapter? You're mostly oxygen. You're just a giant electron gatherer. That's really, unfortunate, but it is what it is. Alright. So, and then magically it made ATP. I did this myself, by the way. You're impressed. Alright, so the inner membrane of the mitochondria, here's the intermembrane space. So here's my drawing of the mitochondria. Here's the outer membrane and the inner membrane. What's this part called? Matrix, and I've drawn that here. So scientists, chopped this thing up and they figured out that it's made of all these different parts. So what I want you guys to do is know the parts. Remember I made you know kingdom, phylum, and so on. So I want you to know complex 1, 2, CoQ, complex 3, cytochrome C, and complex 4. These are made of a lot of different parts that I'm not going to make you know. But you need to know the order that this occurs in. And there's a reason for that. So I'm going to erase all this. And we're going to draw this. So this is the inner membrane. We have complex 1, 2, CoQ, 3, site C. This is that cytochrome, remember, that changed color. And then 4. That's it. That's all you need to know. So what happens is NADH, right, it has an electron. It's been trucking around inside the cell with it. And complex 1 is more electronegative than NADH. So when NADH comes up to it, where does the electron go? Here. And CoQ is more electronegative than complex 1. So where does the electron go? What if CoQ was less electronegative? Would the electron go there? And so you wouldn't get the flow of electrons, right? In fact, all these things in this exact order are more and more electronegative. And what's the most electronegative? Oxygen. So the electrons are going to flow from NADH through 1, CoQ, 3, site C, 4, and on to oxygen. FADH2 holds its electrons more tightly. So when it gets next to 1 cannot pull that electron off, but 2 can. So for FADH2, the electron path is same, except it skips 1. So it goes through 2. So NADH skips 2, FADH2 skips 1. Everybody got it? Alright, so I want you to look at this. Is that integral or peripheral? It goes across the membrane, it's integral, right? Integral or peripheral? Integral, peripheral. Integral, peripheral. you see an integral membrane protein, what do you automatically think? It's going to transport something. And so you would be right. So just a couple side notes. This is complex 2. This is the enzyme succinate dehydrogenase that we studied in the lab. You guys probably forgot that, but remember we did the DCPIP and we made the blue color clear, what we were supposed to, but it didn't really work well. So that's what's going on here. Succinate's converted to fumarate. It makes FADH right here, right? And then it uses it. I asked all the other instructors that teach in this class, and they're like, we don't have that problem. Must be crazy.
Yeah, how long are their classes?
So they're the same. And the only thing I can figure out is, like, because they're used all day, they must just get really hot for my class, and then it just screws us over. Yeah, it's so weird.
Somebody's got to be screwing with you.
Yeah, I don't know. It's weird. I'm like, you got to be kidding me. Nobody else has this problem. Anyway. It transports stuff, remember? So integral membrane proteins usually transport things across membranes because most things can't go across a membrane without a channel, without an integral membrane protein. What can go across without an integral membrane protein? What's the rule? Small? Charged can't go through. Uncharged can. Yeah, hydrophobic. Right. So non-hydrophobic things are non-polar. That's correct. So it's what you said was the same. All right. So just here in the yellow is what you need to know. These are the other parts. I'm just showing you this just in case you wanted to, memorize this for your biochemistry class. But just exactly like I drew on the board, right? It's just showing you in a written out form the way the electrons go from complex 1. It skips 2, goes to CoQ, to 3, to cytochrome C, and then on to 4, and then on to oxygen. FADH2. Complex 1 can't pull it off, so it takes this pathway instead. And so it's like a rock rolling downhill, right? All these things are more electronegative, and if they weren't, the electrons would stop going, right? And we don't want that. We want the electrons to flow, because if I cut the cord on your vacuum cleaner, it doesn't work anymore, right? And if you didn't get the electrons to flow here, what would happen? So you guys know what cyanide is? Do you know how it kills you? It binds to this right here and stops the flow of electrons. So what's the ultimate result? You don't make enough ATP. Being poisoned by cyanide is the same as holding your breath. It's the same effect. Guess what else binds there? Carbon monoxide. Cyanide binds here covalently. Carbon monoxide binds ionically, but they bind at the same place. So which one's reversible? Yeah. So if someone came in your hospital and said, "I've just, eat cyanide," you'd be like, "Sorry." But if they're like, "I just got carbon monoxide poisoning," you could be like, "We can reverse that, right? We can give you oxygen." What? 10 minutes if you get enough. I mean, there's cases where people have slowly been poisoned by cyanide. Remember, you have 100 to 1,000 mitochondria per cell, and you have 1 trillion cells. So it would take a lot of cyanide to bind to every single, and then there's millions of electron transport chains in each mitochondria. So it would take a lot of cyanide to block every single mitochondria in every single cell. But if you don't get enough ATP, it's going to make you weak, and eventually it will kill you. Alright. This is just to remind me to tell you that if if I gave you a plastic fork and a metal fork and asked you to shove it into the outlet, which one would you pick? Why? It doesn't, yeah, it can't move it's not as electronegative, right? So different things have different electronegativities, even forks. And this is just showing you that same pathway that I drew on the board. Electrons go through here, change that, and then end up on oxygen and make water. And then this is FADH2, so it takes this other pathway.
That's a great animation, really.
Are you mocking me?
No.
Fine. Alright, so that brings us to, so this is the mid-70s, right? Scientists know all these complexes, they know where the electrons go, but what is a mystery is, how do the electrons magically make ATP? Nobody knows. And what are they for? Like, what do the electrons do? Nobody knows. So this guy, Peter Mitchell, he came up with this idea that NADH has how many electrons in it to give up? 2. Remember I showed you before it has 2? 2 electrons go through here. His idea was, oh well, electrons, what's their charge? Negative. And what's the byproduct of making NADH? Remember, it's hydrogen, a proton. So what's attracted to negatives? So his idea was, when the electrons go through here, the protons go out. Makes sense, right? Because he looked at it and he's like, those are integral membrane proteins. They got to transport something. So he proposed this theory called the chemiosmotic theory, which says, as the electrons pass through these integral membrane proteins, the hydrogens are attracted to them and they get transported out. So he got sick and he wasn't able to follow up on the experiments, but that was his hypothesis. So you guys knowing what you know, what experiments could you conduct to see if this is true? So if this were true, protons would build up in the intermembrane space, right? What is the result of protons being present? Okay, that's one. We could test the charge. What else? What are a bunch of H pluses? What are a bunch of protons do to a solution? It would change the pH, right? So I know we're going over a little bit, but I just want to finish this thought. So the pH should be lower in the intermembrane space, right? Because we have more protons, and there should be an electrical charge. So his students, just like you guys, did the experiments, and they found that it was true. pH is lower, charge build up. They even took the inner part of the mitochondrion, put it in some acid to see if it would make ATP, and guess what? It did. So in 1978, done a Nobel Prize, $1 million, hunk of gold worth 50,000. Didn't even give any credit to his students. Complete jerks. Yeah. Yeah. Okay. But he won the Nobel Prize before he died, because they don't give the Nobel Prize to people that are already dead. That's why one of the people that discovered the structure of DNA, Rosalind Franklin, never got a Nobel Prize. Alright, so we'll pick this up on Thursday. We'll finish this chapter on Thursday, and then we'll have a review on Tuesday, and then you guys will have your test on the 6th, which is a Thursday. I got was from